It is now, well, we're about halfway through the 24-hour event. Is very exciting. So thank you, folks, for coming on. The next hour is going to be dedicated to multi-scale visualization. Quick note, we had to pre-record this hour because of scheduling conflicts. We have panelists on three continents. Well, sorry, three time zones, two continents, France, and then Eastern time and Pacific time. I do actually see that one of the panelists, Ludo, is actually in the audience. It's very exciting. So we're going to be playing this recording, but I am here available on the on Zoom to answer any and all questions that you might have, and I'm going to do my best for just providing resources and links in the chat as the video goes along. With that, please enjoy this hour. I enjoyed it greatly talking with these amazing people. I had you're going to see the introductions here in a second, but I had the honor of having Tobias Isenberg, Griffin Weber, and Ludo Autin with me on the panel. And with that, Nicole, let's get it rolling. Thank you.
All right, everybody. Welcome to this hour in the Multiscale Human 24-hour event. This hour is going to be all on multi-scale visualizations, and I'm happy to be joined here by a gallery of amazing folks joining me here for this panel today on December 14th at 11 p.m. My name is Andreas Bueckle. I'm the research lead here in the Cyber Infrastructure for Network Science Center at Indiana University. I have with me three amazing panelists who are going to give talks and then join me for a panel discussion afterwards. With me are also Natisha and Meryl, who work here at the center with us and help us make this event go well. Tobias Isenberg, also on the call, is a senior research scientist at Inria at the Saclay Université Paris-Saclay campus, coming to us from France today. Ludovic Autin is a staff scientist at the Scripps Research Institute in La Jolla, California, coming to us from the West Coast, so lots of different time zones here. And then Griffin Weber is Eastern time. He's at Harvard Medical School. Griffin is an associate professor of medicine at the Beth Israel Deaconess Medical Center and also an associate professor of biomedical informatics at Harvard Medical School. So plenty of expertise, plenty of time zones. And with that, I want to give a brief primer to set the stage about building and exploring the Human Reference Atlas with virtual reality. So again, my name is Andy. I'm the research lead here at the center. Our team, and you might have seen that in the preceding hours, has developed a series of user interfaces to map and explore the human body at single cell resolution. One of these interfaces is the registration user interface, or RUI, that allows you to generate a 3D extraction site for a tissue block inside of a reference organ, in this case, the male left kidney. And then the sister interface is the exploration user interface, which is web deployed, which allows you to zoom in 3D through the human body and look at all of the tissue blocks that have been registered for all of the organs. And so what we were doing when we looked at all of this amazing data coming out of this is, well, what if? What if instead of exploring this data on a two-dimensional screen with keyboard and mouse, what if instead we could stand right next to the data and appreciate and explore the organs in their true 3D scale and use some of the advanced immersive techniques for interacting with this, both 3D and 2D data in an immersive scale? And so we implemented the Human Reference Atlas Organ Gallery, which is available for free on the MetaQuest store and which we have also documented in a paper. And one of the important aspects of this organ gallery is its multi-scale nature for exploring spatial data. The starting level for the organ gallery, as you can see here, is the whole human body. That starts at the scale that we are most familiar with as humans, which is about the centimeter or meter scale. But over the course of the life cycle of the organ gallery, we aim to extend the explorable levels across many scales inside of this multiscale system, all the way down to the gene and protein level. We envision the human body for this as an inverted skyscraper, where we start at the top level, which is the whole body level, and where we can then descend into floors below this by virtue of this elevator panel, and where we get kind of sized down by one order of magnitude every time we click one of these buttons. There are 3 other floors available. So we have the whole body floor, which is available. That's the default one. And then we have 3 more available: small organs, large FTUs, that's functional tissue units, and cell groups at the about 100 to 1,000 micrometer level. And so here is a collection of GIFs that show you what you can actually do in this application. So the whole body level is the scale that we are most familiar with. This is the default scale for humans, which is about the centimeter or meter level. In this world, 1 meter in VR corresponds to 1 meter in the virtual world. The user can explore whole organs here, in this case, a whole kidney. And then the -3 level, so being scaled down by a bunch of orders of magnitude, allows you to explore a Visium slide that's 6.5 by 6.5 millimeters. So in this case, a meter corresponds to a millimeter. And then we have this 3-dimensional stepped relief map visible here. At the same level, we have some auxiliary 2D scatter graphs that have been imported into the VR scene here that the user can explore to contextualize the 3D visualization that they can see on the ground. And then one further level down, we have the cell groups level, which allows you to explore single cell data. Each dot here is an individual cell nucleus from a tissue stack for the human skin. There is also a demo video here available that details this particular scene. All right, that is the primer that I wanted to give as far as the Human Reference Atlas and our work in VR is concerned. I want to give it over to Tobias.
Thank you. I hope you can hear me okay.
Yes.
Okay, next. So I want to tell you a little bit about 2 projects that we've done on the visualization of essentially cell nucleus to DNA data. And the first technique was called scale trotter. And here the challenge was like, how do we visualize all the different levels of detail that we can now have data for? Some of it is simulations, but some is real scanned data. How can we simulate or visualize that in a continuous way? So we have the nucleus of the cell, of course, and we have the chromosomes, we have details in the chromosomes, then we have the level down that is fibers, and then comes the nucleosomes, the nucleotides, and the atoms. Next. And in previous techniques, what we did is easy transitions between different levels of detail. So here, this is shown for a molecular or protein structure. Next. And here you can see these transitions, and you see that this is fairly easy because essentially the geometry matches. So one level of geometry matches to the next. So it was essentially a transition of different geometries. Next. But this doesn't really work in our case because these levels of detail are vastly different. They basically don't interface with each other very easily. So this kind of geometry transition wouldn't have worked. Next. So what we did instead is develop a new technique, which we call visual embedding. Next, and that works as follows. So you see this transition, and that is essentially borrowed from old illustrations. Next. Here's 2 examples from Grey's Anatomy, where the illustrators essentially draw a coarser geometry or coarser level of detail, and then the details where we want to see something very much in detail. So essentially, the coarser level provides the canvas for the more detailed level. And that essentially translates very easily to computer graphics. So if we use that now, there's a couple of steps that are happening. Next. So first, we show the coarse level in 3D. Next, then we flatten this out so that it becomes this canvas. Next, then we add the detail of the next level on top with simple alpha blending. Next, and then we basically let the previous canvas disappear, and we are at the next level. And then, of course, we zoom in at the same time as well. So now an example for a little bit larger scene. Next. So here you see that in action. Coarsening, flattening, detail added, and then the final detail is revealed. So in order to show you this continues through the scale, the next slide shows a video that should play automatically. So here you see the cell nucleus, and now we zoom in slowly. The chromosomes are revealed, and there you already actually don't see just chromosome dots, but you see details of the chromosome. So each chromosome has several locations, and then as you zoom in further, we slowly, as they come closer, we start to reveal the next level. And at some point, you have to select where you want to go, onto which chromosome and which detail. Here we selected this beige chromosome, And then again, as you go down, you have to select the next level. So here we pick a few sequences and zoom in. And here, of course, because that's a long stream, we cut this off at some point. And then we zoom in further and we see the nucleosomes already. And now we add the atom structures. First, the nucleobases, and then the actual atom structures. And there you basically see that you can continuously zoom from the outside of the nucleus all the way into the atom level. Next. But the initial inspiration for this work actually was a different one. Those are that you see here from illustrations of all these kind of levels of detail at the same time. And if you look at these are done by illustrators, and they make their life easy. They cheat. Next, if you can, like here, there's a few points highlighted where they cheat, where they kind of nicely hide some changes behind things. They basically also just use the H chromosome, where we want to use this interface representation. So it's easy to create these illustrations, but if you really want to do this with data, this becomes a lot more complicated. Next. So what we do essentially here is two things. The first thing is we want to go from this regular structure to a straightened structure. Next. So for the chromosomes, that's kind of easy. We kind of put them in a line. Next. For the next levels, it's interesting because now we have like a path structure that we want to follow. Next. And here we basically want to straighten each higher level element in a line, and each lower element takes some local information. Next. So we have a straightener. That's kind of the location overall, that those are arranged in a line. And the straightenee next. These are these detail things, and they are kind of interpolated along the way so that they kind of nicely connect as they go on, but keep some local information. Interestingly, the straightenee of one level next is the straightener of the next level, right? So because we are now then one level down. Next. And then for the nucleosomes, it's fairly easy again because they are kind of individual points. Next, so we basically line them up and connect them with some filler DNA. And the final two levels next are straightening for the double helix scale and the basis scale, where we essentially just unroll things and flatten the bases. And then next, for the basis level, we also like spread them out a little bit so that we can see the atoms at some point. And now for the arrangement next, we basically do this in a zigzag fashion where we have constant scale zones and then transition zones where we transition from one level to the next. So I have talked about this like straightening in the constant zones, but I still have to talk about the transition next. And for that, essentially, we could have used, or we used again, yeah, next. We used again the same transition that we used before. Next, so here we had this transition with a visual embedding. We essentially do the same thing, now only on a spatial scale. Next, so essentially what we do is we start with a coarser scale, add detail, remove the coarse scale, and have the detail level there. And that happens for several of these transitions. Next, here we see that for low-key to fibers. Next. For there you see again this correspondence between straightener and straightenee. Next, so one dot is the same as the sequence below. Low-key to fibers. Next, fibers to nucleosomes. Next, and then finally the unraveling of the nucleosome into the actual DNA stream. Next, and then we basically transition to the basis level. And then we have everything, and we can put this together next. So we have one big overview for multi-scale unfolding with the different levels and the transitions next. So we have all the different levels there next. And on the other side, we kind of see the spatial relationship, or the data relationships next. There you see times 25, times 100, and so on. This is not spatial scale, but this is number of elements essentially that you have there. And then finally, we also want to interact with this next. So we want to be able to pan the scales, and that starts automatically too. So here you see that we pan at the lower level, then the next level, and then the next level. And here there's one downside. Maybe we can talk about this at some later point. The speed of motion is not correct here. simply because we cannot render at the needed speed. So whatever I do at the top there, it would basically fly by at the bottom. And I have to admit, next, there's one point. Oh, sorry, I didn't actually have this here in the slides, but I can talk about this later. There's one point where we actually have to cheat as well, similar to the illustrators, but that was needed. Otherwise, it wouldn't have worked. Just wanted to mention that this was work together with my esteemed colleagues from TU Wien and KAUST and the nanographics group that they have there, headed by Ivan Viola. And the students who did the work were mostly Sarkis Saladian here, the first person, and David Kurill, who worked on this project. Okay, thank you. That's my little spiel.
Thank you so much, Tobias. I took some notes and already added some questions, and I'd love to hear how you cheated in the Q&A session later.
Tell you about this.
Thank you. All right, I suggest that we get through the talks first, and then we're going to have all of the Q&A. Okay, so with that, we're going to go over to Ludo. Ludo, are you ready?
I'm ready.
All right.
Can you hear me all right?
Yes.
All right. Well, thank you for the opportunity to share with you guys the work I'm doing at Scripps. So I'm going to try to talk to you about the Mesoscale challenge. So next, on the first line. So I think by now you all know that the human body is by nature multiscale and multi-layer, and is made up of different organ systems, hundreds of organs, made up of different type of tissue, made up of a lot of cells, made up of molecules and atoms. And just to give you some order of magnitude, in one typical human body, you have 30 trillion cells, and a human body of 70 kilograms is roughly 7 octillion atoms. So it's a little more than the estimation of grain sand that you can find in the Sahara Desert. So that's a lot of number. And so what if we want to visualize this? Next. So we have tools and scientific method to visualize most of these scales. From your eyes, you can see the skin and the body, and you can use a magnifier to start to see your cell skin. And then you have microscopes that can help you see tissue organization and organization. And you can go down to organelles with some electron microscope, and even down to the atom with some X-ray and NMR. However, there's an area where we cannot see, or I would say that we can see in something blurry. Next. And so that's what we call the mesoscale. It's between the macroscale and the nanoscale. And so to give you an illustration, here is an image of, I think it's the source of a virus. And you can see that the microscope, and it's blurry. So you have no idea of the distribution and how organized the different molecules and atoms in that cell. Next, please. And so I want to illustrate this problem with a story. So Escherichia coli is a bacteria that was discovered in 1886 by Theodor Escherich, and this is how he illustrated it in his studies. And so he figured out that it was a rod-shaped bacteria, but that's it. It's only the shape of it. Next. And a century later, we have electron microscope that can give us a better idea of it, but still we don't have the detail of what's inside. We have research and scientists, but we don't know yet. Next. And so that's when David Gutser, one of the pioneers in this field, he tried to gather all the information he could find on Escherichia coli, and he put it together in a grayscale drawing. And so he carefully drew every individual protein at the membrane and inside. And you can see that during the year, his drawing and painting involved because knowledge advanced. So like in 2 decades, actually in 3 decades, you can see that it's more and more complex. But we still don't have experimental method to see that. So it's the only one that was doing it. Next, I just want to show you another. Oh, sorry, and it's still only a small portion of the actual big Escherichia coli cells. Okay, so next, and another good example of that problem is here. You have on the left a microgram, again, from what we can see with a microscope of a human cell that is being infected by the shabby. So you can distinguish the shape of the virus of the cell. You can see some organelle. You can even see some protein, like or really big complex protein, like the ribosome, but that's it. You don't see the special organization of other molecules. And to illustrate this, David did this huge illustration at scale of what the current knowledge of structural biology and what is inside the cell will look like. But of course, it's an interpretation that we're doing. And so David has been an inspiration for me and for a lot of people in the lab and all over the field. And next, please. And so what we first did was to develop a tool to let people be David. So, okay, so let's say that you want to be able to make your own illustration to represent a cell, a virus, or anything at this molecular scale. And so we follow the same principle that he's doing. We use simplified shape for the protein. We do only cross-section, and we limit the layer. We only do 3 layers in depth, exactly the way he's doing in his painting. And we're pretty happy with this software because it's been used by some teacher and student, and I think give them a good understanding of scale and how things are in viruses and cells. But this is only in 2 dimensions. So next, please. So with Graham Johnson, back in the day, we developed CellPack, and the idea was to try to do what David is doing, not in 2 dimensions, in 3 dimensions. And it's the same idea. We simplify shape of protein and compartment and virus, and we developed an algorithm that will distribute and scatter protein on the membrane and protein inside and DNA and RNA. And so next, please. With this method, we were able to build the first complete model of HIV mature virus. And that was in 2014. And just for the story, like this model took 20 minutes to build. It took 20 minutes to distribute the protein inside and at the surface. It took 40 minutes to build the lipid membrane with a specialized software called Lipid Wrapper. And it took roughly a couple of hours to render the image. With at the time it was Maxwell Wonder, if I remember. And so it was great, but we were limited, and it was difficult to visualize it in real time, and so we were a little stuck. And so I reached out around, and next, please. And I started to collaborate with Ivan Viola group, and so that's how I met Mathieu Lemizic. Mathieu developed a really incredible rendering engine to display this system in real time. So we could look at billions of atoms just real time. And so that opened the door for me to work with Tobias, and we port a lot of CellPack approach directly on the GPU. And so the distribution was now completely instantaneous. So we just move a slider, it will change the distribution and make the model for us at real-time performance. It was really incredible, and we pushed that with Tobias to link that to some physics engine, so we can see really that things are relaxing, and we can play some game, and we can put some gravity on, and this is to illustrate that even though it looks crowded, it doesn't occupy all the space. You know, it's only 30% of the space that is occupied by macromolecules. So next, please. And so with that method, we had Martina Maritain that was working in the lab, and she applied the method to build a model of a full bacteria, which was Mycoplasma genitalium. So we picked that because it's one of the smallest bacteria that exists. And I put on the side a comparison with, of course, David Goodcell painting from 2011, and our attempt to model that cell with CellPack. And so just for the story, it took me a week to generate the model. And it was only a quarter of the protein and DNA that would have been in the cell, while Martina's model is complete. Next. And actually, because we didn't have the bottleneck on the technology for the rendering and modeling, the difficulty was to gather the data and the information, like all the proteins that are involved inside, what are the membrane proteins, what are their functions, and are we confident in their structure? Because when she was doing this project, it was before AlphaFold. So she had to build every protein by hand. So it was a gigantic effort, and she did a great job, and the model is really great. But the thing is, all of that technology require like high-end computer graphics, high-end software. And so what we did, next please, is I reached out to David Chenau and Alexander Roll. They are the main developer of Mostar, which is a molecular viewer running in the browser. And so I worked with them so we could use their technology to render this large-scale model. Next, please. And so we use the same technology and approach than Mathieu, Dominique, and Ivan, which is level of detail and culling. And so you don't render every atom, you render every over or every tenth. Next, please. You leverage advanced shading that help see the depth, like here is the screen space manipulation. Next, please. And we implemented a clipping object from a plane and cube and cone and cylinder, and that tremendously help when you're analyzing these models that are volumetric. So you need to get through them and look into them. Next, please. And since it's in the browser and it's front-end development, in Mostar, we had the possibility to add labels and to create some interactive session. And so we made available to the user a way to generate a guided tour of their model where they can create snapshots like slides, like a Google Slides. And we can do a guided explanatory tour of the model. And so we're really happy with that. And you can do it for all the models available or your own. Right, I think next, I'm going to get close to the end. And so just some key points to take away. I think for me, the collaboration is key. It's like, reach out to people and collaborate, and that's how we do breakthrough, and that's the best way to go. The data gathering is a bottleneck. Like, where is the data? How do you get it? And how do you handle it? And it's a lot of work. And it is, I think, that level of detail is the common approach for all the multiscale visualization. We use imposter, change representation. So I think this is the key. And then how far are we to visualize the full human cell with molecular detail? I believe that we already have the technology and they are aware, capable of doing it, but we are missing the data. So what you will be looking at, you're not sure what's going to be the truth of what happened in the matter. And I'd like to finish by acknowledging Professor Arthur Autin, which is a big inspiration of all this work, and without him, none of that would have happened. And I found it was appropriate to show you, he's the one in the video playing in virtual reality where he's trying to build the cytoskeleton of a blood cell. And it's just to illustrate the problem we have in scale. So if you work on a membrane at the molecular level, and you're looking at a human cell, like a red blood cell, it's not like looking at a building or a mountain. It's like looking at the sky to the moon or the sun. The distance is humongous. So that's it for me. Thank you very much for your attention.
Wonderful. Thank you so much, Ludo. One of the things I really love about this panel already is just how many visualizations there are. Few words on the slides, many visualizations and animations. This is excellent. Thank you.
Thanks.
Okay, with that, we're going to move over to Griffin.
Thank you so much for inviting me to join this panel as well. Next slide, please. I'm part of an effort with others on this call and many more called the Human Reference Atlas. We're trying to build an atlas of all the organs, cells, tissues in the human body. My role in here is trying to build a multi-scale map of the blood vessels, a daunting process when you look at the picture on the right. Next slide. I'm helped in this effort by Kate Castillo and Avinash Bopana and many others, and the database at our current state is available in a recent paper in Scientific Data. Next slide. So how do we begin to even think about a map of all the blood vessels in the body? And we like to think of an analogy of multi-scale map of roads. So on the left here is a map of the daily traffic on the US National Highway System. The red lines represent the highways, and the thicker they are, the more trucks that are on those highways. It looks eerily similar to blood vasculature networks. And then as we zoom in on the right to a city, Boston, where I work, you can see more and more detail of the smaller streets. Interestingly, the large highway that goes right through our city is often nicknamed the Central Artery. Again, representing that analogy between the transportation network and the biological systems. Next slide, please. So as we further go into different neighborhoods in Boston, each neighborhood has distinctive road patterns based on the history of when those neighborhoods were developed. On the left, we have the Back Bay area where there are lots of parallel streets, and they often remind me of the parallel vessels in skeletal muscle, represented by the green fluorescent lines in that picture. In the Beacon Hill area, there are lots of cobblestone roads. They seem to look to me like brain vessels. And the oldest part of Boston, downtown region, has lots of seemingly random streets, and that somewhat looks like the open blood flow in the spleen. Next slide. We built a video and links down at the bottom where we illustrate this analogy in more depth. Here we're thinking about a truck following rows to deliver a package to a house, and how a red blood cell flows through blood vessels to deliver oxygen to organs. Next slide. So at the highest level, you have the truck going down a highway. A highway could cross 1,000 kilometers. A red blood cell starts out in large arteries, which can be up to 1 meter long in an adult human. And then next slide. As they enter a city or a organ, the truck is moving down towards smaller and smaller streets, and the blood vessel goes from smaller arteries into arterioles. And then finally, in the next slide, the truck pulls into a driveway, and the red blood cell stops or slows down in a capillary where the oxygen is transferred over to the nearby cells. And next slide. So now that we kind of have this understanding of the multi-scale of transportation networks and of blood vessels, how do we actually build that map? And a challenge is that there's no one expert that knows everything about the blood vasculature. There are surgeons and anatomy experts who know about the larger vessels, histology experts or vasculature biologists who may know about the smaller parts, different organ experts. So we need a visual tool to help bring these different domain experts together to be able to document this. So in these diagrams, we draw them out in PowerPoint. It's a simple tool, but everyone knows how to use it, so it's effective for what we're trying to do. The rectangles represent organ structures. On the left are larger gross anatomical structures. And then as you move to the right, you go to smaller organ segments and eventually the smallest functional tissue units on the right. And then we overlay them with lines representing blood vessels. The largest arteries are on the left, and then you can imagine a loop of vasculature going from arteries to arterioles, going through capillaries and returning through venules, veins, and back out of the organ. As I said, there seems to be domain experts who know about the left. You can find those large arteries in anatomy textbooks. And there are other experts that know about the right, the capillaries, functional tissue units, histology. Where there seems to often be a knowledge gap is on that middle. The blood vessels all have to connect into a loop. And if you're trying to draw that diagram and you don't know how to connect up the loop, it means there's a knowledge gap and there's more information that we have to discover. So this multi-scale diagram is helpful for us to document what experts know, work with different kinds of experts, and connect the dots to make sure we know the full pathway of vasculature. Next slide. Here's an example of when we work with the pancreas experts and we draw out the different parts of the pancreas on the left and how it the vessels pass into smaller and smaller functional tissue units. The next slide is showing some more complexity that we see in the liver. And then finally, the brain is one of our most complex vasculature organs, many different components to it. But again, we're still taking that same motif where the largest lobes and structures of the brain are on the left. And as you go towards the right, you're heading toward these small and smaller structures. These aren't visualizations where in real time you're zooming in and out at different levels, but it's important to see all the levels simultaneously so you can identify those different gaps and how the blood vessels connect between the different layers. Thank you.
Wonderful, Griffin. Thank you so very much. Okay, with that, I would like to go over to the Q&A part. I did prepare some questions that we can discuss, but I also wrote down questions for the speakers. So maybe we can first take about 10 minutes to just ask each other questions about the respective talks. And I would like to open with a question for Tobias. So Tobias, you mentioned earlier that in your visualizations, at some point you had to cheat. And I would love, and I mean, cheating is part of making visualizations. Sometimes you have to, cut some corners at some end to make it run on specific hardware, or to jump over some cognitive gaps. I'm curious, what was that instance that you mentioned there?
Well, I mean, I basically in quotations, I made fun of illustrators that they cheated a lot, like in one of the slides. But I have to admit that we needed to cheat in the same way because otherwise it was conceptually impossible. So as we did the multiscale unfolding in the zigzag fashion, right? One of the constraints was that we wanted to see everything nicely in the upper scales, moving left to right. But at the bottom scale, of course, we have the double helix, which we unfolded. So there is exactly like at the bottom, you have this flattened helix, and you move that left to right. That means at some point you are accumulating rotations. So that's easy on the second line from the bottom because that keeps rotating. But if you would follow through with that, basically we would have to turn everything increasingly fast, right? And that's just impossible. And so essentially at the point where the second line curves around from the right into the nucleosome, all these accumulated rotations are basically stopped, right? There, like it turns very fast, but we basically pretend it didn't, right? Otherwise, everything else would be turning as well. But that is just a consequence of wanting to have a flattened DNA structure at the very bottom. So that's kind of a cheating that we had to do.
Yeah, it seems to me like one of the challenges here is that, this is a one-dimensional system, like this is one line. But then because you have the rotation, it kind of adds a three-dimensional component to it, and there might be conceptual clashes.
In a way, the everything is kind of one-dimensional or flattened 1+D kind of representations, except of the actual helix, right? That is a fairly 3D structure. But that is essentially if you flatten one and fix one end, then it rotates. Like if you actually play the animation from the slide, you see as I start, actually that's one further. Yes, the panning. Play that. So at the bottom we start panning it left and right, and you see that the next level was rotating, and now again it's rotating. But this as you move faster and faster, it's accumulating, and that's one of the other things where we actually failed from the graphics side. Now, as you move the upper scales, the lower scales would have to move so dramatically fast that you would not see any detail anymore. So the detail that you see is in a way wrong. You would see just blur. And that's something where I wish we could have continued and explored ways of showing that this vast difference in scale somehow also has to be translated into a visual metaphor in a way, right? And that's something that we don't have in this representation.
This reminds me of what Ludo showed in his talk, which was, being down inside of a red blood cell and looking up at the sky and seeing red blood cells the size of the moon. Just in this case, instead of looking at scale, you look at speed, right?
Yeah, that's a thought that I actually also had as you go down in scale, so looking at smaller and smaller geometries, the processes that are connected to these, which we are usually not looking at all, are becoming faster and faster, right? I mean, you could continue down beyond the atom into subatomic particles, and the speeds are just mind-boggling of processes as physicists understand them right now, right? And that's something that is a very hard problem that I don't really see people have even like scratched the surface yet, right? This time dependence of these multiple levels is really a tough problem.
The time depends, and also like, you know, showing scale differences, that is something that can be done in 1 or 2D, but showing speed differences is even harder to visualize. For scale already, you know, sometimes with the screen state that you have to get to your limits, but with speed, it's even harder. That kind of translates into a question that I have for Griffin. So Griffin, on Tobias and Ludo's slides, we saw a lot of 3D renderings, 3D animations on very particular scales. What you did and the work that you do, what's very valuable is conceptual work, conceptual visualizations, you know, using PowerPoint for showing the blood flow through all of these organs. So I want to talk a little bit about how we can how you think we can unify, you know, conceptual or global visualizations, like, you know, a large scale street map or one of these PowerPoint visualizations, with like more local visualizations that show, for example, the blood flow in arterioles or in capillaries around a very particular organ. I was wondering if you had any thoughts on that.
You know, in the other presentations, a lot of it was you know, video or animation where you're showing the transitions and you're moving between them. And when I'm showing the animations of the truck and the blood vessel going through, it's time-based. When I got to the last schematic diagrams, it wasn't changing. These are static images. So I had to show all the information at the different scales at one time. And that was important because, again, we wanted to see where there were breaks between the different levels. And that's the parts we had to fill in. I didn't want to show the high level and then the next transition to the low level because you missed that whole part. The transition part is the part where we need to capture the information and we don't want to lose that part. So, you know, it was a different approach where I'm not showing the time or so much the kind of flowing through it, but I want to highlight all the different levels simultaneously, so where you could see where there's better resolution and where it was more blurry in terms of the amount of knowledge that was there. So it's kind of, do you show one level at a time and do the transitions between them, or show all the levels at the same time but lose other aspects? Like I lost the 3D aspect of it and some of the other complex parts of it by having to show everything in one static image.
Yeah, and I'm wondering on that note, how can you give a user, how can you allow them to have detail and overview at the same time? So, you know, the local detail is relevant for the particular organ or the particular section of that organ at that time, and understanding where they are with regards to the entire vascular system at the same time. I don't know if there's anything in development on your side yet for that particular challenge.
Well, you know, there's always this sort of split screen version of it where you have the 3D representation on the right and the diagram on the left, and you're going through them together. You know, it depends on the use case and who the audience is. You know, in your RUI examples, when you're trying to use this to map tissue specimens to the organ, that might be useful. Here, what I'm doing is a very specific question to the domain expert, and is, do you know the vessel that connects this capillary to this artery? And in that sense, that kind of simplified schematic is really what helps us get to that point. And the 3D representation almost complicates it a bit. People think of that 3D and they kind of, oh, it must connect through something over here, and that really explicitly putting the pieces together. But when you get rid of the 3D part of it and you're focused just on the transitions between the different levels, that's when the organ experts kind of understand what we're trying to ask from them. So, you know, I understand your question about how do you pull all that together. Sometimes it can create some cognitive overload where you're showing too much information, and we're on purpose removing a lot of the normal ways people think about organs in terms of anatomical structures and that, and say, don't worry about that. We're just trying to build out this one vessel loop and show me the different pieces there. So in our part of it, we're not trying to add those things in. We on purpose remove them to bring focus to the specific question we needed to ask those domain experts.
You made a really good observation there with this, which is that 3D brings an interaction cost and a cognitive cost. So it forces you to adjust your mental model, but it also means that you then have to implement tools to actually help the user interact with the 3D content as 3D content. And sometimes with mouse and keyboard, it's just not as straightforward as it is for interacting with 2D interfaces. There is a question for Tobias in the chat from Kadi. I want to first just ask, or maybe Kadi, why don't you ask your question to Tobias?
Yeah, so great presentation. It's amazing to see all that work and also the synergies across the 3 attempts to help us understand this 10 raised to the power of 10 zoom. It's massive. I wonder if you have tried to show scale bars in 3D. I think Andy is also trying to advance our understanding of how to do this better. And also time scale bars. Have you tried this? Have other people succeeded in doing this? How do we do this together across these different efforts?
In terms of time, I haven't looked into this. I have some sketches that I've done at some point, but I really haven't looked into this kind of visualization. But I essentially liked this idea of this split screen representation, where on the one side you have the actual 3D, and on the other side you have conceptually where you are in scale space. And in a way, you could do that with what I showed, right? You could show this multiscale unfolding on the one side that tells you where you are in scale space, and then on the other side, you could do the actual 3D zoom into things. In a way, I had envisioned initially, if we would have had more time in that PhD work, to do a transition between the two. So at some point, you are in 3D space, and now you focus on one point, and can you transition to the multi-scale unfolding representation? And then you do your further exploration there. And from there, you fix on one point and say, I want to see that point outward now in 3D. So can you come up with a transition that kind of lays out everything as it was in the 3D? We haven't done it. It would be amazing to look into that, but I don't know if it even would work.
Very good. And then follow-up question. So in the Human Reference Atlas cell type population visualizations that Andy is working on, we oftentimes can only show 1 in 10,000 or even 100,000 cells because it's so densely packed, right? If we would put all of them in, then there would be no way to show the rare cell types. There would be no way to see all the structures and patterns, which is densely packed. So how do you do this best, and how do you have then another number, right? So now you have a scale bar, a time scale bar, and then a ratio of how many you can show per packed object. So now you have 3 numbers to deal with. Should they all be in the lower right-hand corner? Have you seen any solutions that work there?
I know that some people in the Viz community have looked into this generation of cell interiors. I mean, Ivan Viola's group is very well known for this kind of work, for example. But I don't know if they use bars to represent these numbers. I mean, what they are trying to solve is essentially a graphics problem that you cannot render all of this stuff. And I think there's the other problem also that Ludo is kind of addressing or trying to address is where does all that data come from, right? Like how do we get all that data and figure this out? And Griffin also talked about this. So as you go down, you need more and more data, right? And where do you also then stop displaying? I mean, you only can view a certain range. So there's plenty of really interesting challenges in this space that still have to be addressed.
Very cool. Thanks so much.
Thank you. I do have a question for Ludo based on his talk. So Ludo, you were showing CellPack and CellPlane, which are amazing tools for the broad community that anyone can use. And when I visited you, it was amazing to actually see how you were building these using Unity. So I'm curious, how can folks, use the results of the cells that they visualize, to use those in papers and other places where illustrations are needed? Like, are there any what are the workflows that you point people to in order to actually deploy the visualizations that they make?
So I think the easiest pathway right now is to just use the regular molecular file format. So instead of downloading a structure or protein, you download the virus, and then you import it in your preferred software. Like if you are an illustrator, you will go to Blender and what's called the molecular nodes, or you can go in VMD or Camera. That's how it goes. But they are really complex systems, and so it depends like how much detail you want from them and what you want to do with them. So because okay, now we can build them. What do we do with them? So we're gonna start to work on doing the simulation, and so that's I can talk a little about this time scale also, because it's all the same problem we have with molecules. You can do a molecular dynamic that will explore time scale of femtosecond for a protein, but we can do Brownian dynamic that will do maybe microsecond. You can do finite element dynamic. And so that's an interesting question. And personally, I would love to work in that. And I know that Ivan with Mathieu Music, they have one paper that try to solve this issue that I really liked, where you slow down or you accelerate depending on where you're looking at. And so you can keep things blurry and fuzzy and going really fast where you're not looking at. And then when you look at something, you can just slow down. And so what I will do is I will use the same way you have you zoom with the camera with like the wheel or anything. You could have a zoom on time. And so you could control at what time scale you want to look at. And that's the only way you could do it, I think. And it's the same for the multiscale. It's level of detail. At some point, you're replaced by imposter, and then you go on a different level. So you can stop with a CT scan and then go down to the cell, replace the cell with some light microscope image, go down and replace with something else, or until you get to the atom and you can replace with the particles. But this can be done very smoothly with, I think. Does that answer our question?
Thank you. Yes, that absolutely does. We have about 10 minutes or so left, and I would like to hone in on three more questions actually. So I'm gonna share my screen here again for the Q&A slide. Okay, so I actually added this question at the very end while listening to Tobias talk at the very beginning, and I want to talk about stepless and stepped zoom. So stepless zoom refers to what you would see in something like Google Maps, where you zoom in with your mouse scroll wheel. I mean, under the hood, it is also stepped, but it appears as if it's stepless. And a stepped zoom is what you would have, for example, on a microwave, when you turn a button and then the time to cook something increments in like increments of 10 or 15. Stepped versus stepless zoom. Both have pros and cons. And I want to start a brief discussion about which of these two are preferable for which type of visualization and for which type of data. And Tobias, I was wondering, since you brought this up during your talk, if you could present your thoughts on this.
So just from the top of my head, I think the continuous zoom, the stepless zoom is something that is very good if you want to teach about something. So you understand what's going on, and you want to tell other people, this is how that thing relates to the other thing. Like if kids have to learn about anatomy in school, that's something where you can show, can zoom in and out almost like seamlessly, and hopefully from that you get an understanding. However, if you need to do a task at a given level, then you need to stay at that level, right? If you need to analyze data at some point. Griffin talked about this. I think that is a very good point where you need to stay at one level, understand what's going on conceptually at that level maybe, and then having different stages where you look at things at different hierarchy levels, but at discrete hierarchy levels.
Thank you. Yeah, I would add to that, maybe it's also about presentation. If you present something to someone or teach it to someone, then the ability to rapidly go in and out might be really helpful. And if you work on something, and also it might be very easy to accidentally zoom to a completely different level if there is not a stepped zoom functionality.
And maybe you can also add, like, conceptually, often like these concepts happen at a given level, right? And conceptually, you cannot do this this continuous zoom in at least some cases, maybe even many cases. And then if you want to understand something at this conceptual level, even if you're presenting something, you need to stay at a level. It doesn't make sense then to continuously zoom. And you can show interfaces as Griffin did. Maybe Griffin can actually talk about this, right? I think that's something that's possible, but conceptually you're staying at then two levels at the same time.
Right. Griffin, did you want to? Yeah. Respond.
Yeah, so the biology of this is that you have large blood vessels, and then there's a branching point where a smaller vessel comes off of it. So the biology is somewhat stepped in that you're going through different branching levels until you get down to the capillary. But in the static diagram where we have arteries on the left and capillaries on the right, we're not focused on just one level. Again, the interesting part is this transition. So on that one slide, even though the individual vessels are stepped, it's almost like a stepless transition that you're seeing the gradual transition from the large vessel to the small vessel and trying to find where the gaps are. And that transition between the big and the small is where we need to have the most time speaking to the domain experts, even though if we were able to zoom in, you can see there are actually steps representing the different branching levels of the vessel.
To round up this part of the discussion, I want to share my screen one more time. As part of our effort to construct this room and reference address organ gallery, I mentioned earlier that we have this idea of an elevator system, which is a stepped zoom. So every time you click a button, you get shrunk by 1 order of magnitude. So an exercise that we did a couple of weeks ago was to go through other major depictions of different scale sizes in science education and try to align the scales. So this is the famous Powers of 10 movie that I assume most of you have seen. And so we tried to take screenshots from the movie and align it with the elevator scales. Now, one of the challenges here is that, and we didn't really notice that until we started doing that. For most levels, the ones that we're the most familiar with, you assume that each of these scales corresponds to one major entity. So, for example, the 10 to the power of 0 meters is just the whole human. But once you get down to a sufficiently small scale, the scale difference, even within 1 power of 10, is so gigantic that it is impossible to describe it succinctly with one particular zoom step. So, for example, 10 to the minus 4th gets us down to the 100 micron level, which we have also modeled here in VR. This gives you down, this overview of the cell stack here. But the challenge with that, at the beginning of this frame, you're still outside of the skin. So you go into the hand of the man, right? So this is still outside. So this is like one of the pores. And then by the time you're about halfway through the zoom, you can see the collagen. And then by the time you're done, you're all the way down here. And this is hugely different from where we started. So even if you have a stepped zoom, there's going to be subscales that you probably have to take into consideration. And again, we didn't even think about this until we, really started to align, and until we started to look through this at the lens of actual stepless zoom, which the movie tries to make, but of course they have to cheat as well, as you might know.
And this is actually one of the very nice observations from that movie. Is that's kind of where they probably first also realized is that the meaningful levels of scale are much more densely packed in the biological world than they are in the other extreme in the astronomy side. Right there, things are very far apart in terms of orders of magnitude. Whereas in the biology, you're packing so much into very few, in quotes, very few orders of magnitude. It's already a lot of orders of magnitude, but you're packing so many levels in there that it becomes a huge challenge how to manage all of these.
Yeah, the level exposed that the movie exposed that very beautifully was just vast emptiness of space, which is terrifying in its own right. Wonderful. It looks like we are about at the time where we have to wrap up. This was wonderful. I could definitely talk for another hour with all of you folks. I will be on the Zoom call answering any questions that might come in via the Zoom chat. Thank you so much for jumping on this call with us. Looking very much forward to the rest of the event, and I wish you all a wonderful rest of the day.